Okay. I hope everyone can hear me. Welcome to the second installment of the protein and protein folding workshop. I guess today we're not as many as last time, but anyways, we're going to continue. First, let me go to the presentation. Okay. So we're going to start off by just reminding you how the setup is, right? Same as last time. So optimally, you're watching on your, you have your laptop with the links open that I sent you over Slack, and you're watching the stream on either a second monitor or a other device like a tablet or a phone, right? I also hope that you have all the files ready and also the program open and the whiteboard open. If not, let me actually go to the whiteboard. Okay, if you've joined the whiteboard, please choose a color, write your name, same as last time. I'm going to be using black. Okay, so you can either draw or type, either one is fine. Okay, very good. Someone else, only see 3 so far. Still waiting. If you don't have the link, please let me know in chat. Who removed me? I hope nobody removes anybody from the whiteboard. I'm not sure if that's even possible. Okay, 4 people. Optimally, if I look at my stream, there are 10 viewers. So optimally, all of you would join on the whiteboard. 5 so far, 50%. Okay. Anyways. Yes. So if it doesn't work right away, take your time. Just listen to the presentation and try to join meanwhile. Otherwise, I'm going to continue. Okay. So, the setup, right? Everything's fine. I wanted to start with a little bit of review today, because surely last time I gave you a lot of information and not all of this probably stuck. So the most important points I wanted to highlight once again so that we can then continue and build on that knowledge to extend it further to understand how proteins work and how they fold. One of the main key aspects that we talked about last time is molecular interactions. And the main feature of all molecules is hydrophobicity or hydrophilicity, I would say. Right? So hydrophobic means they're scared of water. If you would put the substance together with water, it would try to have as little contact with the water as it could. As we know maybe from math, volume per area, the best ratio is found in the sphere. Right? On the other hand, you have hydrophilic molecules or substances, which are attracted to water, right? So if they would come in contact with them, they would try to get as much contact as possible and just spread out or mix with them. Either one could work, right? So last time, if you maybe remember, we were talking about what would happen if we put a peptide, right? So a chain of amino acids into water, right? These amino acids, like we talked about last time, have different properties. Some of them are hydrophobic, some are hydrophilic, some are even have form ions, right? Positive and negative ions. Right? All these have an effect on how these proteins fold in water, because water is their normal environment that they exist in, right? So the environment's usually salt, water, and other proteins. Right, so if we go to the whiteboard, you can see that I've prepared 1 to 4 4 short peptides, right? So the green one is the hydrophilic part, right? The one that loves the water. The red one is the hydrophobic part. So what you would like you to do is, like last time, I would like you to draw what those would look like in water. Right? So just imagination. This is not really an exercise where you could say, oh, this is completely wrong or this is completely right, because, well, folding is somewhat of a oops, okay, yeah. So there's no right or wrong. Just try to basically draw it on the whiteboard as you think that it would fold, right? Let's just start with number 1. Let's see. Who is going to... Any takers? Any ideas? There's a little bit of lag on the stream, so I'll give you a little bit more time, like we saw last time. Sometimes it takes a while for you to be able to respond, so I'm going to give you a little bit more time today. If there is anything unclear about what I'm asking you to do, please ask a question. Right? So once again, I'm going to write down a little bit. So this is, the green part is hydrophilic. And the red part is hydrophobic. Right? To help you a little bit, once again, if it's hydrophobic, it does not like water, so it will try to have as little contact as possible with the surrounding. Someone? No one? Okay, we have one idea. Okay, so in this idea, there's a little bit more of a bend in the hydrophobic part, which is good. Which is a good idea, right? to give you the solution. So probably the hydrophilic part would stretch out, and let me change the color and the size. So one part would stretch out because it wants to have as much contact as possible. Oh, it's hard to draw in colors. Okay. Okay, I see. Well, you don't necessarily have to draw in colors. You could also just draw the structure by itself. Right, and maybe it would be something like this. Right, would like coil up. Oh, very good. On the right, we also have a good candidate. Right? This is also a very good idea, right? In many cases, the hydrophobic part, actually, they bury themselves inside the protein coil, right? So this is a very good answer, as we see here. Let's go back. I don't know, there's a purple box. Okay, very good. So let's go to number 2, right? Now it's not 2 parts, it's 3 parts, right? So we first have hydrophobic, hydrophilic, and hydrophobic again. So what do you think? What would happen if we were to put this peptide into water? Oh, very good. There's a good idea to the right. So that's a good idea. So that could happen. So the middle part stretches out, and the left and the right part, they coil up, right? Like in the other example. Okay, here also, same idea. Very good job. Right. Oh, there's another one in the making. Okay, excellent. Very good idea, right? So additionally to coiling up, they would probably also see contact with each other, right? Because hydrophilic and hydrophilic substances, they like each other and they're looking for as much contact as possible, but also hydrophobic materials or substances, right? They are also looking for as much contact as possible with each other. Okay. Very good, very good. Okay. Okay. Yes, very good. So I would actually like to highlight this one because that's the one in the middle. That's the one I would think would be the most likely to happen. Right? It's not 100%. It's just very likely. It very much depends on exactly what amino acids you have, and also on the conditions in the environment. Okay, going to number 3. So now it's just inverted. So on the outsides you have hydrophilic parts, and on the middle part you have the hydrophobic part. I think by now you get the idea. Okay, so I'm waiting again for your responses. Okay, that looks very, very nice. This one over here. Right, again, coiling up as little contact to the hydrophilic surrounding or to hydrophilic parts as possible. Do we have some other ideas? Very good. Excellent. Another one here. Same idea, coiling up in the middle, and the other parts are extended out. Okay, very good. So now, coming to the last one, right? So I made it a little bit more tricky, and I would give you a little bit more time, and I would like everybody to try to think about how this peptide would look like in number 4. Because this is a little bit, it's trying to make you understand how it actually looks like in bigger proteins. Okay, so try number 4 now. Very good. Here's a very, very good idea. Okay, so as we saw before, the orange parts would coil up, and perhaps also the outsides are attracted to each other. That's very good. It's a very good idea. This one's also a very good idea, although a peptide only has one beginning and one end, right? In this case, you have 6 different ends, so it means you have 6 different, or at least 3 different peptides in this coil that you drew, right? But you have a very good idea. Also, the hydrophobic parts are coiling up and are attracted towards each other, right? Other ideas? Oh, okay, very good. Same idea, but more connected, right? So the hydrophobic parts, they all gather in the middle, and the hydrophilic parts, they stand out towards the outside or the water surface. Very, very good ideas. And the last one is a little bit of a trick question, because actually, to draw this one, there are many, many different options, right? There is not one single correct answer. As before, it really depends on the composition of the amino acids, which exact alignment or which conformation it would take, right? The chain is possible. The hydrophobic core, I would call it, this kind of structure is possible, right? This structure up here is also possible, right? So only like some of them are in the center and some extend out, right? So very, very good. So I wanted you to see a little bit, the more complexer the peptide gets, the more options there are for it to fold, right? Okay. So let's get back to the presentation. Okay. So we just reviewed the hydrophobicity and the conformational changes that happen to it in water, to peptides in water. And also last time we talked a little bit about other interactions that are important within proteins besides hydrophobicity and hydrophilic parts, right? For instance, depending on the amino acids, there can be, for instance, ionic bonds, right? In what you see in the top left. Or, I mean, we just saw the hydrophobic interactions, right? There can be also covalent bonds within proteins. And I would say probably the most important ones are the hydrogen bonds, right? So we talked about last time a little bit that some molecules, they can share hydrogens, right, or protons. And by getting in close proximity, they basically almost form like a semi-covalent bond, right? And these play a very, very important role in proteins. So in this case here, you can see those are, this hydrogen bond is formed by a side chain, but this can also happen between the the peptide bond, right? Okay. Oh, okay, that's what we just answered. So, coming to something a little bit new and a little bit more into detail, right? Perhaps you remember from last time, we talked about what nucleic acids look like when you look at their chemical formula, right? So we talked about why they are called amino acids. And one important thing was they have basically an amino part and they have an acid part, right? If we think about the center of this, if you just don't focus on the green and red for now, the center part is basically one amino acid, right? They're all the same if you look at the bottom part. What they differ in is the so-called side chain, right? And if you remember, there were also 20 different types. So, and they have different properties regarding, depending on the amino acid side chain. So, but what's also important, if you look at not only one amino acid, but two, is basically that actually the connection, what you call the peptide bond between the nitrogen and the carboxylic acid group, this one that actually cannot rotate, right? So usually you have only two different angles in which this molecule can rotate. So because now we're going to step a little bit away from the 2D representation that you know from chemistry, and we're going to go a little bit more into a 3D environment, right? If you think about normal molecules, actually all of them can rotate, right, around in their bond. In this case, this one's limited. Why shouldn't really matter to you right now? I mean, there is a reason, but it's a little bit complicated to explain and would take a longer time. The main point is that there are two axes that this amino acid or this short peptide that consists of 3 amino acids can rotate, right? So this is called a phi, I call it phi or phi bond, and this is a psi bond, or psi. I'm not sure about the correct pronunciation. I'm always going with the Greek, because those are Greek letters. Okay, so basically last time I also told you a little bit that those can rotate, right? And they don't really rotate in every bond, but they only rotate in two bonds that are connected to the central carbon atom, So let's say if you go back to the whiteboard, let me actually go back. In nature, there actually exists not only the chaotic structures that we looked at before, if we think about the hydrophobicity or hydrophilicity of molecules, right? They don't just coil up, but sometimes they build structures. Those structures are basically limited by what I just talked about, by the rotation of the bonds in the atoms. So if you think about it, right? If you take the peptide from number 4, coiling up is one thing, but let's say there's only limited movement in this chain, and it cannot exactly coil up. How would you make a structure out of that with a regular shape, right? So maybe that's a little bit hard for you to solve, but I would actually still like to try for you to think about it, right? If you think about it, if you put if you're making spaghetti, right, and you have a cooked spaghetti and you drop it on the ground, well, it's gonna coil up like we thought about it in before with the hydrophobic parts. But if you think about it, let me go back. If you think about it, oh, all right, never mind. If you think about it as more like a chain, right? So if you look at a chain, going back to the whiteboard, if you look at a chain, I'm going to draw it for you. Right? A chain, you cannot twist like without a limit. Right? There's always basically a certain angle between the different separate parts of a chain that they cannot extend over. Right? So if you would twist the chain, there's only a limit to it. So if you would think about it as not like a wet noodle, but as a maybe a necklace or a steel chain, what kind of structures would you be able to make with it that have like a regular and repeating motif? Do you have any ideas what that would look like? I'm going to give you a couple of seconds to think about it. It's a little bit hard to imagine. If you don't come up with anything, I will give you the answer. Okay. That seems to be a difficult question. I don't know what this is supposed to be, but okay. So I'm going to give you the answer. So as soon, I mean, in the 1960s, I think was the first time we had an actual picture of a protein. And as soon as somebody looked at the protein and looked at the angles in the molecules between them, they made a diagram, right? And this diagram actually shows you all possible structures that can be built with a peptide chain, right? This is a little bit complicated perhaps, but just what the main point that I want to talk about is there are 3 main structures that can be built. One is the anti-parallel beta sheet. The other one is a parallel beta sheet. And the other one is a right-hand alpha helix. Right? So if we look at them in a chemical way, or with a chemical formula, this is what they will look like, right? So these are extended amino acid chains, right? You can see again the C and the R and the O and the N that form the peptide bonds, right? And what you can see is these structures are supported by hydrogen bonds between them, right? So the first one that you see on the left is the parallel beta pleated sheet. Okay. And the other one is the anti-parallel beta pleated sheet. All right. So both of them have a lot of hydrogen bonds in between them, which makes them very attracted to each other and very stable when it comes to this structure. On the right hand, you see a right-handed helix, right? So in this case, it's like a spiral going upwards, right? It's going upwards counterclockwise. And you can also see in between the different steps of the spiral, there are also a lot of hydrogen bonds that stabilize it and make it a favorable structure, right? Okay, very good. So looking at it with a chemical formula is probably a little bit hard to imagine. So what I did is now we want to go to PyMol. I hope you have it open, right? Maybe the last one that we looked at was this one, right? Unstructured H bonds. Right, hydrogen bond in between the unstructured chain. Now we want to look at the structured parts. So all you have to do is click on the scene on the bottom left, which says alpha helix. Right. So again, I would like you to explore, like turn it around and view it from all kinds of different angles, right? So like the view that it has in the beginning, if you click on it, is as you see it, or as I just showed you in the presentation, right? With the hydrogen bonds between the different steps, or the different steps of the spiral, the yellow dotted lines, right? And the amino acid side chains, they're actually sticking out, right? For instance, if you click on this one, this is with a yellow on it, this is methionine, right? So the central C is this one would be the C with the O, and this one would be the N for the peptide bond. And this is a peptide bond, right? Okay, very good. And this is sticking out. And if you turn it, you can see it's spiraling upwards. So if you look at it from the side, you can actually see there's like a hole in the middle. If you can view it from either top down or bottom up, you will see that it basically, in the middle, it's hollow. And all the side chains there are sticking out. Pretty good. All right. I also found an example in the protein that we're looking at that is a better sheet, right? So if you click on better sheet, it will zoom over to the structure that resembles what you saw in the chemical structure that I showed you on the presentation. Right? So we have 2 amino acid chains. Here's actually bend in the end, and then it goes back, right? And there are a lot of hydrogen bonds in between, so it's a very, very stable structure. Okay. Right, so now a question for you. I showed you both, right? So you have PyMol open, and if you look at the presentation, actually, which one of the better sheets is it, right? Is it a parallel or an anti-parallel? So the answer, type it either in YouTube chat or in Zoom chat. Either one is okay. If nobody knows this, I would definitely like to have the answer from Pavel, because he definitely knows. No. Okay, very good. Right, so Pavel gave us the answer. It's an anti-parallel, right? Very good. So in this case, because what you can see in the presentation is basically arrows, right? So parallel going in the same direction, anti-parallel going in opposite direction. Right, if you look at it, because they are folded together, right? This is one peptide that you see in PyMol, right? Which is just folded on top of each other. So if we were to look at it, think of it as an arrow, if you fold an arrow on top of itself, you'll go in one direction, then turn around and go in the other direction. Right? So it's an anti-parallel better pleated sheet. Very good. Right. Let's see. So these kind of structures make up proteins. That's all of them, right? All that is possible for a peptide chain to form is either no structure, an alpha helix, or 2 versions of a better pleated sheet. That's all you find in nature, right? So you have these huge constructs constructs. Right? For instance, if you go to PyMol and you click on everything, right? So this is our whole protein that we're looking at last time and this time. And you see, those are very, very basic parts. They're just like kind of scrambled together. Right? For instance, if you look at PyMol, and this is what you see, right? The left side, you see a lot of alpha helices, right? In red, orange, yellow. And on the right side, you see predominantly beta pleated sheets, which are, in this representation, they actually even show you the direction. So, and there's an arrow down and an arrow up, right? So a lot of anti-parallel beta pleated sheets. Okay. So now, if you look at any protein, these are things you will easily recognize right away, right? And in between these structured elements, you have a lot of unstructured elements as well, right? So for instance, this yellow line. Okay, so going back to the presentation. Okay. So I hope you will remember that. Because, well, those are the simple, what I just talked about are the simple units of that make up a protein. But if you look at the protein, this is actually the one that we're looking at in PyMol right now. This is from a protein database. Actually, if you look at the amino acid sequence of a protein, you can kind of predict what kind of called secondary structure they're going to form, right? So all we talked about right now were secondary structures. Alpha helices, beta sheets are called secondary structures. So this is basically an outline of the whole protein that we're looking at, and you can see all the secondary structures marked in it, right? So you have the yellow arrows as part of a beta pleated sheet, and well, the green parts are what you call turns, so where you would find like the peptide is turning around, and you have the red lines which indicate there is an alpha helix. But still, looking at this map, you would not be able to tell what the protein looks like, right? You can kind of predict what the secondary structures are, because, for instance, here, this sequence of amino acids is likely found also in other proteins and also takes up the structure very commonly. This happens, right? But if you look at it from the outside, like we did in PyMol, it's hard to tell what it would actually look like in the end, right? Because the arrangement of these beta pleated sheets, for instance, each yellow part could be connected to any other yellow part in theory, right? It doesn't have to be an anti-parallel. It could also be a parallel sheet, which you could form with any other yellow arrow, I would say, in this map. Okay, very good. But there are certain structures that people have looked at and have recognized, okay, those are very, very common things in many proteins that we've looked at so far. So they give it basically like a category and some names. Those are mainly called protein motifs, right? For instance, very commonly seen in proteins are helix, loop, helix structures, right? So you have one helix, a loop, then it turns around, and then another helix. You have very, very interesting structures called coiled coil, right? Where basically well, this kind of looks like DNA if you want to think about that way, but those are actually two alpha helices that are twisted with each other, basically, right? Like a coil. You also, for instance, have things that are called a helix bundle, right? So many alpha helices together, right? That kind of group up together. If we look, well, actually, let me do that later. So we also have, for instance, a beta alpha beta unit or a protein motif, right? So one part of a beta pleated sheet, an alpha helix in between, and then another one. You have a hairpin protein motif. You have a beta meander, right? So where it goes up, down, and up. You have a Greek key beta pleated sheet, which is found very often, where it starts in the middle, goes up and down, and then on the outside goes up and goes one time around with an unstructured loop and then goes down again. Or then you have, for instance, also the possibility of two beta sandwiches, beta pleated sheets on top of each other, called a beta sandwich. Okay, so the question for you, I would like you to look at PyMol, right? And go to the scene that says everything, right? In the bottom left. Let me show you really quick. This is what it should look like. And you get there by clicking everything on the bottom left in PyMol. So if you look at it, I would like you to answer, if I'm going to show you again the different protein motifs. What kind of motifs do you see? Right? If you want, actually, the ones that you find, please type them in either the YouTube chat or in the Zoom chat. Right? So try to look at the presentation, look at the structures, and then look back at the protein and tell me what you find. All right, turn around, look at it from all sides. What do you see? Very good. We have our first right answer. All right, definitely. There are a lot of helix loop helix structures. All right, for instance, especially if we look at the left part. Right, we have, for instance, the very orange helix, which has then a loop, and then another lighter orange helix. Any other one? Any other ideas? So there are two more that you could see in there, or actually three more as an option. Better sandwich, kind of. Okay, yes, actually I would agree with you. There are parts of a better sandwich. Yes, you're a little bit a step ahead, Pavel. That's not a structure yet I'm talking about. So another one that you can find if you look at the red until yellow part, right? This is actually a helix bundle, right? So we have a bunch of different helices that are stacked on top of each other, right? So these are called a helix bundle. Okay, Ashta, very good. There's also a hairpin structure, right? So for instance, if you look at the very green part, so you have one part of the better plated sheet going up, and then it turns around and goes back, right? This is E, the hairpin structure. If you're very creative, or like, if you have good imagination, I would say, you can also find a beta alpha beta unit in there. What I'm talking about is the very light blue part, right? On the opposite of the orange and red part, there's a light blue helix, and which is connected on both sides to a beta sheet. Yes, okay. You cannot see my PyMol because I wanted to show you the structures so you can find out which it is. But if you would like to see it, what I'm talking about is basically this part, the hairpin structure, right? So you have the blue helix on the outside, basically, of the protein, which is connected here to a light blue beta sheet. And on the other side, it's also connected to a little bit more further away, a darker blue part of a beta plated sheet. Very good. And one that you can definitely also find, let me go back to standard, is, if you look at the presentation, the beta meander. Right, going back to PyMol, the beta meander maybe is best visible also in the blue part, right? It goes from, let me see, the dark blue goes down, then around and goes up again, goes around and goes down again, right? And then turns into a helix and goes in part of another beta pleated sheet. Okay, so those are, I would say, the motifs that you could identify. So going back to the presentation, those are things that are very, very common in many proteins. Also, not only like the smaller motifs, there are also bigger motifs. Especially something that I would like to introduce to you, because it looks kind of pretty in my opinion, is something that is formed by a long row of beta plated sheets, right? So in this case, you have like 8 different basically beta pleated sheet parts, and they can arrange in different ways to form a barrel structure, right? And I would also like you to look at this in PyMol, because I've highlighted this once. I mean, you can see it perhaps in the overview when you look at everything, but you have kind of 2 beta barrels in here, right? If it's hard for you to see, click on the bottom left where it says beta barrel. There's only, so I hid all the other amino acids and only showed you the ones that are part of this beta barrel, right? So you can see those are many, many basically layers of this better plated sheet, and they arrange in a circle around each other. And in the middle, you can see there's a hole, which is hollow, or filled with side chains. That might also be a possibility. Okay. Going back to the presentation. There are also, so you might be asking yourself, okay, what's the point of these motifs, right? Why do proteins have those? Because in many cases, actually those motifs have something to do with the function of the protein, right? So certain motifs can have the same kind of function between different types of proteins, right? What I'm showing you right now in the presentation is one example of a helix turn helix motif that we looked at before, which we also saw in the protein. And this one is especially good at binding to DNA, right? So if you have proteins that can attach to DNA, you very, very often find these helix turn helix interaction sites, right? Or these motifs in them that can interact with the DNA. Because if you look at the presentation, especially this helix fits very, very nicely into the big groove of the DNA. I mean, okay, we haven't talked about the structure of DNA yet, but you might know it's like also like a spiral. There's a bigger and a smaller groove, and it very, very well fits into the bigger groove of the DNA. Right? So different proteins with the same motifs also usually or sometimes share similar functions. Right? So we've looked at secondary structures, like bigger secondary structures, but there are also even bigger like structures that are composed of secondary structures, and these are usually called domains, right? So if you look at the presentation right now, you can see, for instance, in the top left, the first one, it's just a bunch of alpha helices that are like scrambled up together, right? And somebody looked at it, recognized it in many different types of proteins, and called it an alpha horseshoe, right? Or somebody looked at other proteins and like we did, right? Like we're doing in PyMol right now, and they recognize, okay, oh, there's actually an interesting structure. That's a barrel that's made of alpha helices, right? In number 3. So you have something that's called an alpha-alpha barrel. Or a 5-bladed beta propeller. Like they all get very complex and very more and more abstract names. Yes, but something that I want you to remember, like the bigger structures of secondary, made up of secondary structures, are called domains. And they usually have their own names. Sometimes they're also called after proteins, sometimes they're called after what they look like, or sometimes very like structural, a 3-layer beta sandwich, right? So there's a lot of freedom in there. Also these, in addition to the protein motifs that we talked about, also these sometimes share function, right? So for instance, like beta barrel, if you think about it, if it has a hole in the middle, it might be able to let something through, right? So maybe all proteins that share this structure, they're able to form like a gate for molecules to let through or not let through. Right? But I hope we can look at that in the next workshop. Right. Or another example, similar to what I just talked about. Sometimes you have domains that, for instance, specialize or are common in proteins that are found in the cell membrane, right? So these are either like bundles of alpha helices with a lot of hydrophobic residues or amino acids that are then able to anchor themselves in the membrane because of their properties, right? Or even they form like a gate, like I said before, and are able then to control what goes through or what goes in and out of the cell, right? Or in the very right, you also can see, for instance, an example for a beta barrel that can kind of do the same, right? So not a small one like we see in our PyMol, but a bigger one that is also to control basically the flow of molecules in and out of the cell. But there are many, many other functions. And perhaps we'll look at different ones in the future. Also, something that I want to go into detail a little bit is, I mean, when you think about it, what you've studied in school so far, I hope what you've been taught so far about enzymes or proteins is what you see on the left, right? So proteins or enzymes are able to catalyze reactions, and they basically have what you call a substrate, something that fits into them, and then they make a reaction happen, and then it goes out, right? I hope most of you have seen this kind of picture. So this on the left is a very, very simplified schematic of what is happening in real life. Right? So, but what's important is to remember these names, right? So I want you to remember a substrate is whatever is binding to the enzyme or is being basically worked on. The active site is basically the binding site for the substrate, something that's specifically made for this to fit into and to be worked on. And the enzyme is basically the scaffold around the active site to allow access and then make the reaction happen. Right? So if you look at the right part, you can see an example of that, right? So again, the schematic on the left, right? Part A and part B, they go into the enzyme and then are bound and are being changed, right, chemically. So in reality, or in our model as we could see it on PyMol, here you can see the one molecule on the left on the very top, and the smaller one on the right. And the binding pocket that is specifically made for them so they can fit in there and they're attracted to it is highlighted in blue, right? And once they're bound, you can also actually see that the structure is a little bit changing. It actually closes above it if you look at the blue highlighted area. And then the reaction can happen. So not all proteins have an active site, but those that have a chemical function as a catalyst, all of them do, right? There are some other proteins, for instance, collagen, or for instance, keratin, right? So structural proteins that you might have heard of, like for instance, when it comes to hair, or for instance, how pretty your skin is, or your fingernails. Those, for instance, do not have an active site. They do not have a chemical function. Yeah, so basically that's what I want to say. They don't have an active site, but some of them do. And if you look actually at PyMol, I have highlighted something for you. Everything parts. Okay. So if you go at INH, so the last one of the scenes in PyMol, I want to show you something, right? What you see here is called an inhibitor, right? We will talk about, I think, more in detail what an inhibitor is in the future workshops. For now, I would just like you to see that this is a molecule that is designed to bind to an active site and not let go, basically. Usually in active site molecules go in and go out again. But inhibitors basically are for the most part designed to fit in there in the same pocket as the normal molecules, but bind in there irreversibly, so they don't go out again, right? So if you look at it, you can see everything, the protein that we were looking at before is grayed out, and everything, the inhibitor is highlighted in gray. Right. So, and what you can see is basically this is the active site. So I actually don't have a scene for that, but you can do it by yourself to make it look similar to what I showed you in the presentation. If you go to the right part of PyMol, you have the different selections, about a little bit last time. So you can choose the protease, and you have the different letters on the right next to it, the A, S, H, L, C. And I want you to click on Show, or S, and then in the drop-down menu, click on Surface. So this is what it should look like. Right, so this is basically showing you the surface of the atoms of this protein, right? And it kind of connects it to look like a cloud, or it's actually hard to describe, right? So the protein that we're looking at actually has an active site, which in this case is blocked by an inhibitor, right? So, actually, I don't know, Pavel, do you know what this protein is just by looking at the active site? If you get to know, or if you work a lot with proteins and 3D models, you can actually recognize the function of a protein by looking at the structure. So, of course you can do that, but also for me it's difficult, but perhaps. What's TF? Transcription factor? No, what's TF? Okay, no. Transcription factor would also not have an active site, I would assume. This is actually a protease. Actually, let me go back. So, all right, actually, let me continue with the presentation, and I want to tell you about this protein a little bit in the end. So I will want to make it a little bit of a surprise. So what have we been looking at this whole time? But let me get to my last point first. So far, we've looked at primary structure, like just the amino acid sequence, the secondary structures with which it assembles into, like alpha helix or sheets. We've also looked at what you call a tertiary structure, right? So domains, motifs, or whole proteins even, that are folded like this, a combination of these secondary structures, and they can form like megastructures. There are also quaternary structures. This is like the final step, like an assembly of different peptide chains that are folded up in a tertiary structure, which then form a functional complex of different what you call subunits. Right? In this case, for instance here, we can see this is hemoglobin. Hemoglobin, I think you've heard about it. This is the part of your blood that is transporting the oxygen throughout your body, right? So in as a model, it will look something like this. It is made up of 4 similar subunits, which both of them carrying a heme unit which can bind an oxygen, which then assembles into like a 4 subunit structure, which you then call hemoglobin, the protein, right? So protein can have more than one subunit or more than one peptide chain, right? In this case, the one protein hemoglobin is made up of 4 peptide chains. Okay, we did the analyzing of the protein. Okay, so ta-da! So what we've been actually looking at is a part of the coronavirus. Like, oh my god. Yes, so this is basically the main protease of COVID-19. A protease, if you don't know it, we haven't talked about the functions and the categories yet, but a protease is a protein that can basically cut other proteins, right? So also what you see right now in the presentation looks a little bit different from what you maybe see in PyMol. The reason is what you're looking at PyMol is basically is 1/2 of it, right? The blue area is the active site, right? What we looked at last with the gray longer molecule sticking into it. But the functional organization of the protease, it has 2 subunits which bind together in almost like a heart shape. Right. When it comes to the function of this protease in what the coronavirus does, it's a little bit complicated. It's also not 100% understood. It has a lot to do with making the cells form new coronavirus particles, right? So what it does, it basically cuts off ends of other proteins, which then makes the cell membrane form little dents, I would say, which then can form new coronavirus particles. So in the thing, though, what I wanted to show you, why looking at 3D structures is relevant, is because we can then, if we know where the active site is and what it looks like, we can then design inhibitors, right? So that, what I told you about, something that is blocking the active site. It basically fits right in there, into the same pocket as the actual substrate, but it doesn't go out as easily, right? So basically, if here what you see on the right, this blue molecule is in the active site, it blocks the normal substrate of this enzyme, and it's not able to work anymore. This is how a lot of medicine works, right? You might not know it, but a lot of the medicine that is commonly used, for instance, like painkillers, those are inhibitors for certain proteins, right? Traditionally, we used to kind of trial and error test them, right? But now we're actually able to look at the proteins, at the structure, and design them, right? Okay. Let's see, what's the time? I think we can continue a little bit. I'm not sure. Maybe actually, I think for today, that was a little bit of enough input, I would say. I don't want to make it too much at once. So let me sum up what we did today and what we're looking at. Maybe by looking at PyMol one more time. So today we were looking basically towards the step between how does a peptide, like we understood from last workshop, how does this peptide basically make huge structures that we can find and identify in proteins, right? So we've looked at the main basic composition that we can find in many proteins, called, for instance, alpha helices. We looked at beta-pleated sheets. We looked at protein motifs and other structures that are typically formed within proteins. And we looked at proteins as a whole and how their structure can mediate certain functions like enzymatic activity or binding and other types of interactions like regulating transport. Okay, so for next time, I'm planning probably to talk more about folding, right? Because if you think about it, we started today looking at or thinking about how proteins would fold just on a piece of paper. But as we saw, there are tons of different possible outcomes for a single peptide. So how do actually proteins assume the same shape with the same function? Right? That's a very important part, because you don't want to make nonsense proteins. You want to have something in the cell that actually can perform certain tasks. So we will be looking at that next time, how that is actually happening. And I'm also planning on telling you a little bit more about what kind of functions proteins actually can have, because this is also a very interesting part, which is important to understanding basically how the world works, how we work, how our body functions, how we can survive a pandemic and other things. Okay. Any questions from anyone who's watching? So if you have a question, either type it in YouTube chat, Zoom chat, or you can also ask it on Zoom. Are all proteins structured? I mean, depends on how you define structured. There are certainly also proteins that are completely what I would call unstructured. For instance, if you look at the protein that we looked at today, the protease, like the yellow part, things like that. So structure is not a necessary property to be called a protein or an enzyme. Any other questions? How do you tell the difference between parallel and intermittent structure for the protein again? Okay. Let me go back in the presentation a little bit. So the difference between parallel and antiparallel is basically the direction of the amino acid sequence. I haven't told you before, but actually, if you look in an amino acid sequence, it has a front and a back part, I would say. Well, front and back is maybe not good. In science, they called it an N and a C terminus. Right? It is basically the direction in which it is made. So usually the front is called the N terminus, right? So it's called an N terminus because it has the amino part first, like we talked about in the. Oh wait, let me show you the presentation. Sorry. So actually, when the end part is first, right? As you see in the chemical formula, and the C terminus is basically the carboxylic acid, which is then basically after the N. So it goes from N to C to N to C, NC, NC, NC. Right? So this is how you would draw this arrow if you have a peptide sequence. So in this case, one peptide, which is on the left, this one goes from top down, right? From N, and the bottom is C, right? And on the other side, this one is an N, and also goes down and ends in a C. Right, so also a downward arrow. If you look at the middle one, the left one starts in N, ends in a C. So downward arrow, and this one starts with an N, and basically starts with a C and ends with an N. Right, so in this case, it goes not from top down, but bottom up. So the direction of both peptides are opposite of each other. That's why you would call it anti-parallel. Right? Probably the more common one is the anti-parallel, right? Because if you would just think about a long peptide, it just could form an anti-parallel sheet very easily by just folding onto itself. Right? Whereas a parallel sheet is usually made up of peptides or sequences that are further apart and then come together and looking in the same direction. I hope that answers your question.